Start.
Good morning. I'm V. Manonmani, Assistant Professor of Department of Biotechnology. So today we are going to say about diagnosis of Mycobacterium. So generally we all know what is Mycobacterium, which comes under the bacteria phyla, and it comes under the order of Actinobacteria and family Mycobacteriaceae. So this Mycobacterium specially causes tremendous serious diseases in humans and mammals. So this complete genus comprises of two different species. One is tuberculosis, another one is lepre. So this tuberculosis commonly causes TB, which we know that this is tuberculosis, which causes a serious cold inside the lungs and cavities. And the next one is lepre, which causes the leprosy in human, which is a skin disease and skin disorder, which usually happens in human beings. So this Mycobacterium, which means in Latin is myco means fungus. We all know what is myco, which is a fungal biology, is called as a mycobiology or mycology. And the next meaning of myco, which means wax. This particular Mycobacteria produces the wax compounds. It is like a shining components which is present inside the media. So that's what we call this wax compounds which is present in the bacteria is called as a Mycobacterium species. So coming to Mycobacteria staining cultures. To view the morphological structures of this Mycobacteria, we have to stain in different staining methodology, which is a common methodology is acid-fast organisms, and the next staining method is rhodamin stain, Gill-Nielsen stain, and Fate stain and Quignon stain. Normally, this Mycobacteria phenotypically is closely related to Nocardia, Rhodococcus, and also Corynebacteria. This Nocardia and Corynebacteria commonly produces serious diseases, mainly the skin diseases in human pathologies. So coming to the main point of this presentation is tuberculosis. So tuberculosis is nothing but a common TB method and which always present in normal air atmospheric levels. The spores will be surrounding in us. When we have a low immunity, this particular pathogens will enter into our tracheal system and it will affect both the lungs. Finally, we will be in a fatal diseases. So coming to the stages of different tuberculosis, the first one is primary tuberculosis, the second one is secondary or cavitary tuberculosis, and the third one is miliary tuberculosis. So starting from primary tuberculosis, normally the Mycobacterium tuberculosis spores will be present in a normal atmospheric level. When the human is at a low immunity, it will enter into the nasopharyngeal region where it enters into the trachea, and it will colonize into a mass-like substances which will filled with the spores of Mycobacteria, and that spores are completely called as gone complex. This gone complex will affect only one part of the lung. Then under the two criteria, it will enter into the secondary tuberculosis. Before entering into the secondary tuberculosis, let me discuss about the rapid or progressive primary tuberculosis. So when the gone complex is less than 10%, we will be getting the progressive primary tuberculosis. Mostly this primary tuberculosis will take place only in children. So under the age of 12, the children will be affected with primary complexes, and it can be easily treated with 6 months and 6 courses of tablets. So and also this progressive primary tuberculosis will get into the immunocompromised host, which means the immunity level is at a certain level of 30%. So this is the two possibilities of taking into the patients of progressive primary tuberculosis. So coming to the secondary tuberculosis, which is also called as a cavitary tuberculosis, this gone complexes will get reactivated or reinfected, and it will infect all the lung parts, and it will enter into the alveolar region where it will block the respiration process. When the patient does not consult at this stage, then it will enter into the miliary tuberculosis. This gone complexes will be divided into certain stages, and it will enter into the bloodstream of the human, and by the capillary system it will taken into the whole body of the human being. First it will affect the lymph nodes, where the lymph nodes will totally disrupt the lymphatic system of the human being. It will enter into the spleen, kidney, and also the bone marrow. Finally, it will go to the meninges, where it is present in the outer layer of the brain. This meningitis will be completely reinfected with the tuberculosis spores, and finally the person will be dead. So this is the 3 stages of tuberculosis: primary, secondary, and also miliary. This secondary is also called as a cavitary because this gone complexes will fill the pleural cavities, and it will be infected with the Mycobacterium tuberculosis spores. So let me discuss about the common diagnosis methodology for this tuberculosis. The first one is medical history, and the second one is physical examination. Third one is microbiological studies. So under microbiological studies, we have to take the samples of sputum, alternative sampling, and PCR. PCR is nothing but the polymerase chain reaction. And the fourth one is radiography. Under radiography, there are 2 types of X-rays. One is chest X-ray, another one is abrevography. The next one is nuclear medicine methods. In this nuclear medicine methods, we'll be giving the radioactive isotopes in which the person is allowed to swallow the particular tablet and then the sputum will be collected. The next one is tuberculin skin test. Under tuberculin skin test, Mantoux test, HIV test, CDC classification of test and BCG vaccine will be given and then the tuberculin skin test will be taken place. The last one is laboratory test. Under laboratory test, adenosine deaminase test, nucleic acid amplification test and also the full blood count, which means complete blood count will be analyzed for the patient. So let me discuss briefly about this diagnosis methodology. The first one is medical history. So under medical history, the patient will be taken to consult the doctor. So there are two stages of symptoms. One is preliminary symptoms, another one is systematic symptoms. So under preliminary symptoms, the patient will be asked for prolonged cough or sneeze or cold which is present for more than 4 to 5 weeks. So that's the preliminary symptoms. The second one is systematic symptoms. In this, commonly the patients will be getting continuous fever, chills at nights and fever at the late nights, and then he will be taking severe sweats. So these are the systematic symptoms of humans. So this medical history will be taken from the patient and the doctor will be editing about this medical history. The second one is physical examination. So under the physical examination, the patient will be allowed to expel the sputum from us, and by eye observation, the doctor will see whether the sputum is greenish yellow or yellow in color. So that's the physical examination of a patient. So the next one is microbiological studies. So under microbiological studies, the sputum will be expelled from the patient and it will be swabbed on the particular media of Mycobacterium. The particular Mycobacterium media is Lawson-Jensen strain. So under that, the media will be completely based in agar media and the swab will be made on the media. So this is the colonies which are found in the LG media. So this is the colonies. You can see the colonies are very shining, which means it's a virulent species, and also it produces a wax-like substances. So this is the confirmation report for the Mycobacterium tuberculosis species. So coming to sputum analysis, the sputum will be taken for staining methodology. After culturing from the plate colonies will be taken for staining methodology. Under staining methodology, mostly Zeal-Nielsen and Orament-Rhodoment stain will be taken place for characterizing the morphological structures of the particular bacteria. So this is the Orament-Rhodoment stain. Here you can see some of the orange color particles. This is nothing but the Mycobacterium tuberculosis species, in which this Orament-Rhodoment will enter into the DNA of the particular bacteria and it will stain completely inside the chromatin structures. So this is the final confirmation report of the morphological views. So next. So coming to the alternative sampling. So under alternative sampling, apart from the sputum analysis, gastric washing, serology swab, broncho swab, fine needle aspiration and tissue biopsy will be taken for the particular patient. Gastric washing, which means gastric washing completely from the bowel movement, and laryngeal swab, a long swab will be taken inside the throat and there they will collect some of the samples. Next one is bronchoscopy. It is also like endoscopy, in which the scopy methodology will be enter into the laryngeal phases. The next one is aspiration. Aspiration, which means from the bronchoscopy, they will enter aspirator needle into the lungs and they will collect some of the fluids. The last one is biopsy. Biopsy is nothing but a tissue sampling from the lungs and they will enter into different stages of biopsy. Thank you for watching.